We want to start to look at the structures and properties of our common amino acids. They can be easily grouped into categories based on the nature of the functional groups in their side chains. Let's start by examining amino acids that carry uncharged side chains. Nonpolar amino acids are included in this group. They're classified as nonpolar based solely on the nature of the side chain. Remember, all amino acids are polar overall. Those that carry an aliphatic side chain include alanine, valine, leucine, and isoleucine. They're represented here in their structural formulas in the center of each figure, the ball and stick model on the left, and the space-filling model on the right. By considering the space-filling model, you can get a better idea of their relative size. Remember, there is a range of nonpolarity based on the extent of the surface area of the nonpolar region. This means that leucine would be more nonpolar than alanine. Glycine and proline are also aliphatic and therefore nonpolar. We saw in our last lesson that these are unique for different reasons. Glycine is the smallest of our amino acids and achiral since it doesn't really have a side chain, just a hydrogen atom. Proline has an aliphatic cyclic structure and is our only amino acid. Another subgroup within those amino acids that are nonpolar is comprised of those amino acids that have aromatic side chains. Recall that a hydrocarbon is aromatic if it contains a cyclic structure with alternating single and double bonds. In phenylalanine, pictured on the left, the ring is a simple benzene ring, whereas tryptophan on the right has an aromatic indole ring. Another nonpolar amino acid with a unique side chain is methionine. It carries a sulfur atom, which you may recall is an electronegative element. However, this sulfur atom is bounded by aliphatic hydrocarbons, and it is this feature that makes the side chain nonpolar overall. Another class of amino acids that have uncharged side chains are those that are polar neutral. Please note that though these side chains are uncharged, these are a class distinct from those that are nonpolar. These are very polar due to the nature of the functional groups they carry. Serine and threonine both carry hydroxyl groups bonded to aliphatic hydrocarbons. If we look at the structure of tyrosine on the left, we notice its aromatic benzene ring, and we might be inclined to consider it another nonpolar amino acid. However, note that it carries a hydroxyl group at the para position on the ring, and this we classify as polar and uncharged at physiological pH. Cysteine on the right is the second of our amino acids that carries a sulfur atom in its side chain, but in this case it is an end-thiol group and therefore polar. Glutamine and asparagine are the last members in this group, and each of these carries a terminal amide group in its side chain. The distinction between them is the number of carbons in the aliphatic portion of their side chains. Why not begin now to review these amino acids, their structures, names, and abbreviations? It will be very helpful as we proceed in order to see how they influence the structures and functions of the proteins of which they form a part.